Hey all, we're today going to be looking at B1.2, which is proteins, and we're going to be focused on the theme of form and function. So how does the structure affect its ability to do its job? The questions we're going to be thinking about are what is the relationship between amino acid sequences and the diversity in form and function of proteins, and how are protein molecules affected by their chemical and physical environment? So firstly, we want to think about what is a polypeptide, and a polypeptide is going to be a long chain of amino acids that fold to form proteins. And in order to form a peptide bond, we are going to have a condensation reaction that occurs between the carboxyl group and the amine group of amino acids. So the amine group is this NH2, and the carboxyl group is this C double bond OH group. So carboxyl groups are acidic and amine groups are basic. And so amino acids are amphiprotic, meaning they have an acidic and a basic end on them. There's 20 different amino acids, and they can be joined together in any sequence to form millions and millions of different combinations of polypeptides. And so here we can see the bond that's formed through that condensation reaction is going to be between the carbon and the nitrogen. And we should be able to draw similar to this, a simple condensation reaction between two amino acids to form a dipeptide. This is called a dipeptide. All organisms use the same 20 amino acids, and that suggests that all life evolved from a single common ancestor, because we're all evolutionarily using the same amino acids, which means they must have come from somewhere originally. So how do we make a protein, or how do our ribosomes make a protein? Proteins are coded for by genes, which are segments of our DNA. And amino acids are going to be assembled together by ribosomes. So in the cytoplasm of the cell, firstly, a strand of DNA is going to be copied into messenger RNA. And we'll look at that more when we look at DNA and RNA. But RNA then exits the nucleus. It connects onto a ribosome, and the ribosome reads that messenger RNA as a code to bring the correct amino acids, and then links those amino acids together. If a polypeptide is 400 amino acids long, it's going to have 20, because there's 20 different amino acids to the power of 400 possible different variations in a sequence. So you can see that there's a lot of variation there. Proteins may contain multiple polypeptide chains, and proteins can be denatured by high or low pH, dependent on the protein, or high temperatures because of the disruption into R group interactions on the amino acid. And so here on these amino acids, you can see the R group. R group just stands for, it could be any real combination of things. So this might just be CH3, but it could be a C2H4. So R groups can be different varieties of extensions off of the amino acid. There are 9 essential amino acids, and what the word essential means is that they must be consumed externally by the body. So these 9 amino acids that are essential are for humans, which means we need to eat them. Otherwise, the other 11 our body can synthesize itself. And the proteome is how many proteins the body can make. It's much larger than the genome. The genome is the amount of genes that we have, but due to how proteins are processed when they are made in the endoplasmic reticulum and the Golgi body, the proteome can be much larger. The way that different functions of proteins, they can catalyze reactions the way that they would with enzymes. They can cause muscle contractions. They can provide strengthening, like ligaments. They can affect blood clotting, transporting of oxygen and nutrients. They can be used for membrane transport, and then also for hormones and neurotransmitters. They can be used for receptors, and then also they're used for packaging up the DNA. So these are histone proteins. And lastly, for immunity. So antigens are a type of protein, so are antibodies. So if you are SL, this is going to be the summary for you that includes a tiny little snippet of HL stuff in here that we're also going to talk about. But proteins are made by genes and through the assembly of different combinations of amino acids by ribosomes through a condensation reaction that will produce peptide bonds. This is the HL component that we're going to talk about in just a minute. There are primary to quaternary structures of amino acids that allow the creation of fibrous and globular proteins, which will have different characteristics. And the proteome is the proteins made by your body, and that's going to be different in each individual. So one TOK question that is interestingly related to this topic is what constraints are there on the pursuit of knowledge? And how do we preserve ethics in human trials? So especially when we're thinking about human bodies and how we're testing on that, these TOK questions do become quite relevant. So HL students, going forward, protein structures, we need to know about the primary, secondary, tertiary, and quaternary structures. So some of our amino acids are hydrophilic, they're polar. Some are hydrophobic, they're nonpolar. And this is going to affect their interactions with other R groups. Some tissues contain a 21st amino acid, which is one of the original 20, but it's modified after the polypeptide is made. So primary sequence is going to be the sequence of amino acids, and that's going to be bonding between the carbon and nitrogen. But this carbon-nitrogen bond allows rotation in that bond like this. So what that means, and that's what this image is showing here, allows rotation so proteins can fold into any shape. Continuing on, the secondary structure of protein is going to be hydrogen bonding of peptide backbones. So that's going to be hydrogen bonding between carboxyl and amine groups because they're both polar. And that's going to cause folding into a repeat pattern. And that could look like an alpha helix like this, or it could look like a beta pleated sheet, which is like this here. And so this is our primary structure. These are our secondary structures. And then a tertiary structure is the folding of the polypeptide due to R group interaction. So ionic bonds between charged R groups, covalent bonds between sulfur atoms, that's called a disulfur bridge, hydrogen bonds between polar R groups, hydrophobic R group interactions. So the molecule will push away and rotate away from water. And so it's going to be the folding of that polypeptide chain. And then lastly, quaternary structure is going to be when multiple polypeptide chains are going to be interacting together. So it's going to have similar interactions as the tertiary structure. It's going to have covalent ionic and hydrogen bonding happening. But then there's just multiple polypeptide chains here. And so this is a good example of this is in hemoglobin. So protein conformations, one example of a protein conformation is collagen, which is a fibrous type of protein, or it's a fibrous protein, and it has a primary structure and a quaternary structure. So there's a sequence of amino acids, and the sequence of how the amino acids are lined up prevents the polypeptide from forming an alpha helix. And that means because there is no alpha helix, then the molecules can line up and attach to each other so that the long fibers can be wrapped together and they will not be soluble in water. This is one of the characteristics of fibrous proteins. Globular proteins, i.e. hemoglobin, have a primary, secondary, tertiary, and quaternary structure. Some are soluble in water because the hydrophilic R groups are facing towards the outside, so towards water. And actually, just to go back a little bit about collagen, there's 3 amino acids in a collagen strand. And one of those amino acids, because of its R groups, prevents it from making the alpha helix. The other one has, one of the other ones has a specific R group that slots together. And that allows them to line up the way that they have. All right, and that is all of the information for this topic. Thanks for watching.